So the last speaker of this session is Todd Evans, Evan Todd, so, from Weill Cornell Medical College. Yeah, okay.
Hi, everybody. It's a great pleasure for me to have a chance to discuss with you a recent finding that the genome has the capacity to sense levels of sphingolipids and respond to activate a transcriptional program to modulate cellular sphingosine levels to maintain homeostasis. And this is a program that really hasn't been described previously. This work has been done in the ongoing context of our trying to understand the role of sphingolipid signaling in development, which I would argue is a relatively understudied developmental signaling pathway, probably because it's a vertebrate-specific signaling pathway, and so it really didn't benefit from our colleagues in the building early genetic screens in worms and flies as much as some of the other signaling pathways have. But fortunately, it's very well conserved throughout invertebrates, from zebrafish to people. And this has been a collaboration with my colleague Tim Pla, who's an expert on sphingolipids. And all the work's been done, that I'll describe was done by a co-mentored postdoctoral fellow named Karen Mendelsohn, who you can see here in a photo taken a couple days ago. Karen is an expert in genetics and husbandry, and pretty much that's why I'm giving the talk, but she should get all the credit for the work. Now, to initiate sphingolipid signaling, you require metabolism of sphingolipid precursor molecules, primarily sphingomyelin or ceramide, to generate eventually the active, bioactive lipid mediator of signaling, sphingosine-1-phosphate or S1P. Now, the only way you can make S1P is through phosphorylation of sphingosine by one of two sphingosine kinases, sphingosine kinase 1 or sphingosine kinase 2. Now, once you've generated active S1P, it can interact with any of 5 G-protein coupled S1P receptors. Now, those receptors are then internalized and activate a host of downstream classic G-protein coupled receptor signaling pathways, controlling cell proliferation, survival, migration, and cell shape. And this has probably been best studied in the context of vascular biology, whereby S1P signaling controls tip cell migration during angiogenesis, and particularly vessel stability and integrity to control processes such as egress of lymphoid cells out of the vessel into tissue. Now, the role of S1P signaling in development was actually discovered rather famously in zebrafish genetics back in the early initial Boston screens from Mark Fishman's lab with the discovery of the miles apart mutation. In the miles apart mutation, the cardiac progenitors, which are labeled here with a myosin light chain in situ hybridization probe, the progenitors fail to migrate as they normally should to the center to form the cardiac cone and initially the primitive heart tube. Instead, they stay out miles apart from each other and will actually form 2 small hearts, which is termed a cardia bifida. Didier Stanier really moved this field by cloning the mutant gene and showing that it encodes normally the S1P receptor 2. Now, Stanier's lab and others, including Kawahawa, went on to show that another gene, 2 hearts or spinster 2, when mutated, exactly phenocopies miles apart. And spinster 2 encodes a transporter molecule for S1P. And what's believed is that in a pretty remarkable signaling pathway that spinster 2 will deliver S1P to the receptor, S1P receptor 2, which is expressed in the endoderm. And that controls convergent extension of the endoderm, which directly or indirectly then leads to a collective cell migration of the mesoderm progenitors in a process that was beautifully described yesterday by Debbie Yellen which she described to us also involves PDGF receptor alpha signaling, and that's needed for forming the primitive heart tube at the midline. And so we started out by trying to ask two questions. First of all, what's the source of the S1P signal? Where does it come from? How is it generated? And second of all, are there any earlier developmental signaling functions for this pathway? It's a little bit difficult to ask genetically in terms of the receptors because there are, as I mentioned to you, 5 receptors, and of course in zebrafish it's even worse, and there's 7 of them. And so it's a bit of a challenge. So we took another approach. We said, well, instead of trying to genetically analyze receptor signaling, let's focus on the kinases, because there's only 2 kinases. And that's true in zebrafish as well. And in fact, there's only one kinase that's expressed during early development, and that's the SPHK2 kinase. The SPHK1 isn't particularly expressed at detectable levels until somatogenesis. So our strategy was, let's just make a mutant in SPHK2. That should eliminate the capacity to generate any S1P, because the only way, as I told you, that you can make active S1P is through phosphorylation of sphingosine by these kinases. And so we did that. Karen targeted the SPHK2 using talons and generated a number of alleles, and the one that I'm describing to you today is a 5 base pair deletion that leads to a frame shift, an early stop codon, and effectively deletes the entire kinase domain, and we anticipate should create a null kinase. So we generated these mutants, generated heterozygote lines across those, and we found that the embryos were perfectly normal, mutant null embryos, supposed presumed null embryos. But of course you'll remember, actually I didn't focus on it, but you'll remember that there's a substantial maternal component of SPHK2, and so we had to generate null females and null males. And when we cross those, 100% of the embryos look like this. They have a cardiomyopathy, they have a cardiobifid phenotype, they even have a characteristic tail blistering, and they precisely phenocopy the miles apart mutation, mutant. Except, of course, the miles apart is not tolerated as homozygous null, and so a quarter of the het crosses generate this phenotype, whereas 100% of these maternal zygotic mutants look like miles apart. Now, this phenotype can be rescued either by the maternal component, which is laid down from mom, or by a single male zygotic allele. So the maternal component is not essential, but it's sufficient. Okay, so this can also be rescued by injection of mRNA encoding SPHK2 into the embryo, or by injection of mRNA that's targeted specifically into the YSL, and that's sufficient to rescue the phenotype. So we think we answered the 2 questions that we set out to answer, which is that it would appear that the S1P ligand needs to be generated in the YSL, which sort of makes sense because that's where spinster 2 is thought to function, and that the way it's generated is through function of the SPHK2, the kinase. That can be provided maternally as a message or zygotically during early development. And indeed, it would appear that there is no earlier developmental signaling function for S1P than it was revealed by the original miles apart mutation. However, just because there's no earlier developmental function for S1P, that doesn't mean that there's no earlier developmental function for the kinase. Because when we looked a little bit earlier, we saw that in fact there is a phenotype in the developing maternal zygotic embryos prior to the miles apart phenotype. And that's a significant delay in epiboly. So you can see that in the wild type when you're around 80% epiboly, in the maternal zygotic mutant is only about 50% epiboly. And so we wanted to just confirm that in fact these embryos were really depleted for S1P. And so we did lipidomics, and we confirmed that indeed the maternal zygotic mutants have undetectable levels of S1P, but we noticed another thing, which is that they also had excessive levels of sphingosine. And that's perhaps not too surprising because lacking the capacity to generate S1P from the precursor sphingosine, you could imagine that that could lead to an accumulation of the precursor product. So the question is this phenotype caused by lack of S1P, or is it caused by excessive levels of sphingosine? So that's pretty easy to test. We've added S1P to the cultured embryos, and we found that we were unable to rescue the phenotype. But when we added excess sphingosine to wild-type embryos, we phenocopied the sphingosine kinase 2 maternal zygotic mutant. Now, these embryos will survive because they'll catch up, and they'll go ahead and eventually form the miles apart phenotype. These embryos will survive also, and they'll catch up, and they'll go on to form normal embryos. However, we found that if we added a little bit more sphingosine to these wild-type embryos, they didn't survive. They had a rather dramatic phenotype, which is that they essentially exploded. So at about 50% epiboly, what happens is that the blastoderm pinches off from the yolk cell, and this happens relatively quickly. So Karen has to actually just watch and wait until it starts to happen, and within about 5 minutes, it pinches off and the embryo is gone. This occurs by addition of sphingosine, regardless of if it's a target for the kinase, actually. And it doesn't occur by adding excess ceramides, short-chain ceramides, longer-chain ceramides, or S1P itself. But it does occur if we add small molecule compounds that have been described as being inhibitors for sphingosine kinases. And as an aside, it also occurs if we use a morpholino to knock down the kinase, too. Now, what's really more interesting is that this occurs in wild-type embryos, as shown here, with pinching off of the blastoderm from the yolk cell. But it doesn't occur in the sphingosine kinase maternal zygotic mutants. So they will tolerate excess levels of sphingosine and go on and develop perfectly normally. Now, if we keep adding more and more and more, eventually they will show this phenotype. But there's a nice window whereby this is essentially another phenotype of the maternal zygotic mutant. They're resistant to the excess of sphingosine. Now, why these embryos undergo this process is pretty obvious in that there's a total disruption of the actin cytoskeleton that is attached to the blastoderm and anchored at the vegetal pole of the yolk cell, shown by phalloidin staining. There's also disruption of the microtubule network, and this process actually was described beautifully yesterday by Ashley Bruce. If you saw her talk on it, basically it helps pull the blastoderm down through epiboly. The actin network that's present around the circumference in the YSL actually seems to still be present, and so the blastoderm will pinch off because it's failed to be brought down around the yolk cell. But the question is, why doesn't this occur in the maternal zygotic mutants? And so we thought about that, and we realized that in the presence of excess sphingosine, how can you deal with that if you don't have the kinase to move sphingosine through to S1P? Well, perhaps you move back up the metabolic pathway. And so that would mean really two ways: moving sphingosine back up into ceramide. And so we did more lipidomics. And indeed, we found that the maternal zygotic mutants are accumulating long-chain ceramides much more than the wild-type embryos. Now, there are really two ways that you can imagine this could occur. This could occur either by downregulating ceramidases so that you don't accumulate as much sphingosine, or it could occur by activating expression of ceramide synthases so to move sphingosine back up into ceramide. And so we looked at all the enzymes that are encoded in the zebrafish, of which there's quite a few that can control these pathways. And we found that there was exactly one gene that was changed in expression, which is called ceramide synthase 2B, which is highly upregulated, actually as a maternal transcript, only in the maternal zygotic mutant, but not compared to wild type. And none of the other enzymes are changed. And if we knock down expression of CERS2B in the context of the maternal zygotic mutant, we phenocopy the exploding embryo phenotype, but there's no effect in the wild type embryos. So we've generated a CERS2B mutant. It's been crossed down to the SPHK2 mutant, but now we need to make it maternal zygotic mutant, so we're almost there, but not quite to validate this phenotype. Okay, so SIRS2B is a very large gene. This is not really drawn to scale. There's a very large first intron, and then a non-coding exon. And then you only go up about 2 kb before you run into the next gene just upstream. And so we just, on a lark, essentially took this about a 1.7 kb element upstream of the transcriptional start site, hooked it up to a luciferase reporter, and injected it into wild type or maternal zygotic mutant embryos. And indeed, expression of this reporter is elevated 10, 15-fold compared to when it's injected into wild type embryos. So the capacity to respond to these higher levels of sphingosine is retained on this promoter fragment, similar to the endogenous gene. So this is convenient because it allows us to carry out essentially promoter bashing and try to identify cis-elements that are responding. And so we've done that. Karen has done that, and she's found 2 elements. One which is really right at the very end of the promoter in the first 50 base pairs or so, which contributes part of the activity, and then probably a second element, which is about 100 base pairs further down, which contributes most of the rest. And it's not shown here, but if she just takes the 400 base pairs that represented here and hooks it up to a minimal promoter, that's fully sufficient to achieve this activation in the presence of sphingosine. Okay, so what I've told you today is that there is a pathway that we didn't recognize, and we discovered this because the SBHK2 maternal zygotic mutants do lack S1P, but they add another feature, which is that they have enhanced levels of sphingosine. And these mutant embryos are able to tolerate this because they found a way, which is a transcriptional activation of the salvage pathway through the SIRS2B gene to deal with excess sphingosine and protect them from potential toxic effects of sphingosine. Our knockdown experiments so far suggest that that's an essential transcriptional pathway, activation of SIRS2B. And SIRS2B gene itself has what we're calling sphingosine response elements, which either directly or indirectly somehow through transcription factors are leading to activation of SIRS2B as a sensing mechanism for excessive levels of cellular sphingosine. I'll thank my lab for a lot of input, but really all this work was done by Karen in collaboration with Tim Ha, and more recently some contributions from a graduate student, Suveg Pandey, and a summer student, Frank Carilli. I'd be glad to take any questions.
Interesting talk. Can you get the upregulation of the ceramide synthase if you dump sphingosine on the embryos? It wasn't clear to me.
I'm sorry,
If you dump sphingosine on the embryos, can you get upregulation of the ceramide synthase?
Yeah, no.
So
not yet. It may be the timing. I think that it's possible, though, that, and this is something we have to explore, is that there may be an essential sensing component that's taking place during oogenesis, and that's needed to initiate it. Now, the promoter still responds, but when we just add the sphingosine, we're not seeing upregulation of SIRS2B.
Todd, my question is somewhat related, I think. I was curious about which tissues you think are actively upregulating. So you think it's in actually the gonad, or
I think it's in the gonad. I think in one thing that I think is really interesting that I didn't point out is that this sphingosine kinase 2 functions just like sphingosine kinase 1 to generate S1P, but sphingosine kinase 2 is predominantly nuclear. And so it may be during oogenesis that it's really important to sense and control nuclear levels of sphingosine to protect the germline. And that's a special role for it, and that may be where it's functioning most critically.
And do you see is it possible to see other zygotic activation, tissue-specific zygotic activation of SIRS2B or?
Right, so we're trying a lot of things, even in mammalian cells, to see if this is conserved, and if we can look in different cell types, endothelial cells and so forth, and we're trying things but haven't got there yet. Great, thanks.